Oh, count to 10? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
Thank you so much.
Okay.
No, no, you're good.
Before you play, just go over here. And I just want to hear if you're coming out.
Is that working?
That's what I'm going to check.
Let me get out of your way.
Now. There they are. Okay. Good to hear.
So we're good to go.
Alright. Should be good to go. Oh, there it is.
That's what it is.
Play it.
And then you can get back to your presentation.
Yeah.
Just to escape. So if you stand over there and just Thank you. Good evening. Welcome. It's so nice to see you all. This is a great pleasure for me to introduce the talk this evening. My name is Cheryl Peach. I'm the organizer of this series. Harry sends his greetings. He's unable to be here this evening, and so you get me. We're going to have a couple of quick logistical things for you. Many of you I know who come to these lectures regularly may or may not realize this is the last one of 2024. We will gather again. I hope to see most of you in February of 2025 when we'll start a 4-talk series that will go through May. So we hope to see you then. Please look out for email missives from our communications department that will tell you about that lecture series, and you can sign up, and we'll see you in the new year. This is really the last in our first foray into doing a slightly different type of production. As you know, we really have tried to change exactly what we're doing here so that it becomes a little more accessible. And so we're going to allow the introduction not to be a typical academic introduction, but something that's done through video and storytelling. But I do want to mention that our speaker this evening is a dear friend of mine and also an extraordinary individual who is actually an alumna of UCSD and Scripps Institution of Oceanography, and now is a director of a very special program at NOAA called the California Cooperative Oceanic Fisheries Investigations. And it just so happens that this is the 75th anniversary of that program. It's truly an iconic program, and I think you'll enjoy learning a lot about it and its history from our very own Dr. Noelle Bowlin. So I'm going to ask Noelle to come up and introduce herself through her video.
I really believe in the mission of NOAA, and it's to serve the people and support.
The Jeffrey D. Graham Perspectives on Ocean Science lecture series features engaging presentations on research conducted worldwide by scientists from Scripps Institution of Oceanography at UC San Diego. Scientists at Scripps are exploring Earth's mysteries and hundreds of research projects underway on every continent. Learn about science at Scripps and join researchers on their paths to discovery.
I really believe in the mission of NOAA, and it's to serve the people, it's to protect the environment, and I think that's really important, while also understanding the importance of the need to sustainably exploit natural resources. And so I want to help with that mission. I am Noelle Bowlin, and I am the NOAA Director of CalCOFI. NOAA is the National Oceanic and Atmospheric Administration, and CalCOFI, that is the California Cooperative Oceanic Fisheries Investigations, a mouthful. It's an ecosystem monitoring program that actually has 3 leads, and I am the NOAA director, which is a federal branch of the Department of Commerce. I believe the first time I realized I was very interested in pursuing science, I saw that when I was in middle school. I grew up in a suburb of Los Angeles. January 1994, we had a very, very big earthquake. The epicenter of that, my childhood home, I think 1.1 miles from the epicenter. And that earthquake flattened everything I knew, essentially. There was not an untouched portion of my neighborhood or my town. My middle school crumbled in many sections. And we were out of school for a while. When we came back, my science teacher decided to have us focus on the earthquake and really look at tectonic plates. I really grabbed onto that because the earthquake was so confusing. When it's out of nowhere, the ground is shaking and everything around you is shaking. And I was a kid, so it was very confusing. I didn't understand, why was this happening? Why did my town fall apart? And my science teacher said, well, we are going to answer that with science. We're going to explain what's happening in the earth and why this happened. I really loved that there was an answer. There was a real answer to why something like that happened. And that was it for me. From then on, I was always very interested in answers to questions in the world around me. And that always led me back to science. But when I hit biology, I was like, oh, it's all coming together. This is it. I went to the University of California, San Diego for my undergraduate degree, and I pursued biology. I was able to retain my NOAA job while I went to school at Scripps Oceanography to get my PhD in biological oceanography. I loved it. I still love it. I am a biologist now, many years later, when I'm not sciencing. So when I'm not here at work, I spend a lot of time with my family. I have a young son. He's 6.5. We do a lot of fun things together, like dance videos. For myself, I really like to exercise. And one of the ways I do that, I like to play volleyball. It's something I did competitively when I was young. My brain quiets down. My brain's always going. But when I'm playing volleyball, I'm very focused on volleyball. It's like when you meditate, and it gives you a chance to get centered and focus and to let the other thoughts just pass by. I don't have to stop them, but they just pass, they wash over, and I get to focus on me and what I'm physically doing instead of analyzing what I'm doing. I just do it. I love it. It's really fun. I'm pretty excited that I get to talk about this, but I get to be a part of this program. I spent my entire career in it, and I think it's really important for people to understand the utility of this program and how it serves the people of California and where it got its start. Because it started in a place where the researchers were trying to answer one question: what happened to all the sardines? Because the sardine fishery crashed Central California and Monterey. Probably many people are familiar with John Steinbeck's book, Cannery Row. And that is the crash of the sardine fishery is the backdrop of that book. But that is the foundation of the CalCOFI program. But it has evolved. It has evolved so many times over, and it continues to evolve. It came from a single question, and it's evolved into an ecosystem monitoring program that's serving all of us in so many ways. And we're at the precipice of this new frontier in the development of offshore wind energy. What will that mean for us in terms of changes to our environment? How will it affect our fisheries? How will it affect not just commercial, but recreational uses of the ocean? CalCOFI is well poised to help answer some of those questions. So going from sardines to that, to wind energy, a lot of
evening together to listen to a little story about this very big program, a very cool program that I get to be a part of, as I said in the video. So I'm Noelle Bowlin. Hi. And I work at the Southwest Fisheries Science Center, which is just over there. I'll show you a little map in just a minute. But first, let's talk about what we were going to talk about tonight, a little preview. Let's talk about a little bit of background so to make sure we're all on the same page for those of us in the room and whomever is online. Hi, Mom and Dad. And then a little bit of history about the program, because the history, I know but it is quite interesting. And then after we talk a little bit about the history, a little bit of back and forth, because there's some fun little tidbits I wanted to throw in there for everybody. Some little touchstones that I think that we all can treat as touchstones. And then talk about today. What are we doing today? And then we'll talk a little bit about the specifics of one aspect of some of the research that goes on within CalCOFI. And then we'll come back to talking to today again, and maybe some musings on where we might be headed in the future together. So let's get to it. Like I said, make no assumptions. Let's just all be on the same page. It's never a bad idea to start at the beginning. So I work for the National Oceanic and Atmospheric Administration, NOAA. NOAA has 6 branches, and I work for NOAA Fisheries. And I think that it's pretty cool that NOAA Fisheries is charged with the stewardship of the nation's ocean resources and their habitat. We are charged with making sure that there's productive and sustainable fisheries, safe seafood for all people to eat, and that we make sure to help with the recovery and conservation of protected resources and ensure healthy ecosystems. I mean, just a really short list of things that we can knock out. Together, though, together we can, of course. So where are we? So NOAA Fisheries is pretty special. Our science centers are co-located with major research universities for a very good reason, to foster awesome collaboration, which is what we do. So the green arrow, so let's start with the orangish yellow arrow. That's where we are, those of us in the room, that's where we are at the Birch Aquarium. And this is the greater Scripps Oceanography campus, and underneath that green arrow is where the NOAA Fisheries building is. So co-located, excellent collaboration. So now we get into a bit of the history of CalCOFI, and it's intricately tied to the Pacific sardine, a noble small fish. It's not a very long-lived fish, but it is quite pretty. And up until size for a coastal pelagic species, and they are quite tasty. But not just to us. They are tasty for all types of predators, and this is one of the reasons they are such an important species in our ocean, not just here in the Pacific, but in many other areas of the world. And so we humans see them as commercially important for all kinds of reasons. We put them in pet food. We reduce them to oil. We dry them. We grill them. We do all kinds of things to consume them because they are such an excellent source of nutrition, packed in a nice, pretty package. But we, like I said, we aren't the only ones that think that. They are ecologically important because there's so many predators that depend on them as an important part of their diets. And this cartoon here is just a few examples of some of those predators. And so again, history. It's so interesting that during World War I and World War II, there was a really high demand for a shelf-stable, easily packaged, high source of fat and protein that could be sent abroad to sustain our troops. And so you got can labels. This is a can label that you see right here on the screen, A New Hope, to really get the people behind the idea that we can support our troops by sending this awesome food source. It was one of the ways. And so fishing pressure really increased during this period of time, and landings were incredibly high. You can see the sardine catch here in blue. And shortly after fishing pressure really increased, you see a very rapid decline in the fishery, in the landings off of Central California. And this had majorly devastating repercussions for California's economy. And like I said in the video, it is the backdrop. I mean, it was, so important, and it's woven throughout so much of California's early culture that it is the backdrop of this quite famous book by John Steinbeck. And so we fast forward through a lot of things, and we get to CalCOFI. CalCOFI was established 75 years ago by a question. It was a question of what happened to the sardines. Was it overfishing? Was it environmental change? Was it both? Was it a combination? And three partners came together. What would become NOAA Fisheries was the Bureau of Commercial Fisheries at the time. What would become the California Department of Fish and Wildlife and Scripps Oceanography all came together, the super duper powerhouse, to tackle this question. And so, but it wasn't just those three research legs of our wonderful three-legged stool. It was also industry. Industry said, well, we need to know what's happening. This is our livelihood. And so they took the initiative upon themselves and imposed a self-tax to help fund a research plan. And so this plan was developed to figure out what happened to all the sardines. This included monthly cruises, at times utilizing 3 ships working year-round to collect plankton, water samples across this expansive grid. A ship going to every one of those dots from the California-Oregon border to the tip of Baja. And so this was an incredible amount of work because there was basic demographics of sardines were not yet known. We didn't know that yet as a community of scientists. We didn't know. And so these ships were sent out to collect, like I said, water samples to understand the physics of the environment that these animals lived in, and then also plankton, where the baby stages, the earliest stages of sardines were living, because we knew something about how fishes made a living, but it wasn't just that. There were also laboratory studies to describe the developmental stages that you see here in these line drawings, and then as well as the behavior of the adults. Adults were caught when they could find them and brought into the lab for study to understand their behavior and to really get an idea of the adult demography. The fisheries sampling was happening as well. So it was this approach to figuring out how to answer these questions by coming at it from different angles. But while doing this, it wasn't just we come up with a method and we keep going. It's, well, this method seems to work, but could we refine it? There was ongoing research during this time to develop new instruments and techniques to enhance data collection and analyses. And so that was ongoing. And so this multifaceted approach, this comprehensive research plan as it was, became known as the oceanographic approach to fishery research, what we now call an ecosystem approach. And so here's one of those little touchstones that I feel like everybody this can resonate with everybody. So as CalCOFI is out there collecting data, there's also science symposia for scientists to come together to talk about what they are finding and seeing in the ocean. Are there any leads on the sardine question? Well, this is when researchers, and you can read this in the publication, the CalCOFI reports publication, when researchers realized through studying how the ocean and atmosphere interact and how the biology is also interacting with the physics, that it wasn't just this local situation that was happening. This El Nino was a basin-wide issue. And so this was first discussed in a CalCOFI symposium, which is pretty interesting and pretty cool. And in the reports, and this is when I'm sure so many of us have read some, the way that scientific literature used to be written was at times more literary than it might be now, at times. And so if you read in this report, it's quite beautiful. By the fall of 1957, the coral ring of Canton Island, in the memory of man ever bleak and dry, it was dry, was lush suddenly with the seedlings of tropical trees and vines. Thanks, El Nino. It's pretty cool. So this was ongoing, okay? And so that was then. This is what the survey pattern used to look like. It was extensive and it took a ton of resources and a lot of people. And so utilizing the information to understand things like, okay, well, if we continue surveying every month for so long we can figure out when spawning is occurring, when peak spawning is occurring, because we did learn that they are serial spawners, meaning sardines can spawn all the time, but they do have peaks in their seasons. And so these are the types of small things that we're learning that absolutely provide super duper important information for fishery management purposes, because this started as a fishery management program. But along with the pattern that you see here, that we say that it evolved, so did the entire program. And so let's focus a little bit on what today is. And that is, instead of this monthly situation on the left, we now, and I should say there have been many iterations of the sampling domain and frequency, some changes over time, but for about the last 40 years, that red box on the right has been maintained. And so we call that today, in the last 40 years, we call that our core pattern, if you will, and we sample that 4 times a year. 2 of those 4 times in the winter and spring, when we have quite a number of the species that we collect in the terms of fishes hitting their peak spawning, we extend that pattern north. And so in the winter and spring months, we extend that pattern north, and we to try to capture the highest density of the peak spawning of some of these species. And so we go out, we still collect plankton, we still collect water, but we also collect a number of other things. And while we're on this, before I click away, you'll notice that there's some color coding in those circles on the map on the right, and that's to show you that the pattern that we maintain today has sampling stations in both international, federal, and state waters. And also on this map, you'll see that there are other areas of interest, like the National Marine Sanctuaries. And so there's quite a bit of sampling that we do. We also incorporate coastal sampling that wasn't always maintained. And so there's a lot that happens on CalCOFI. But what does the sampling effort look like now? And so it's quite a number of things. There's visual observations, looking for mammals and birds. We still do, like I said, we sample for the water using the conductivity, temperature, and depth sampler that has bottles attached to it, as well as sensors to collect information on the physics of the water and the ocean. We have our net tows to collect the plankton. And then we also, with modern day ships, we have underway data that provides a whole host of other data streams. And then we also have environmental data, which I will talk about in just a bit, but there's quite a bit happening. Before we continue, I'd like to zoom in for a minute. I want to zoom in on what comes out of those, some of the things that come out of those net tows, the plankton or ichthyoplankton, because there's also fishes in that plankton. And so this is where, like I said in the beginning, the 3-legged stool of Scripps, California Department of Fish and Wildlife, and NOAA Fisheries. NOAA Fisheries focuses on the ichthyoplankton that comes out of the CalCOFI samples and has since the beginning. And so let's go back to that, because this piece of the story is so critical to how we've become who we are today as a program. And so the original focus, as I said, was on sardines, and particularly sardine larvae. The babies are a lot easier to catch. As you'll notice in this image here, there aren't robust fins that make this a very strong swimmer. So when a net is coming, it is hard for this little individual, this is 4 millimeters long, by the way, to swim out of the way. And adults can swim out of the way pretty quickly. In some of our other surveys, we also, as part of the Southwest Fisheries Science Center, we collect adults, and that requires a whole lot of other equipment because they're good at swimming away, but the babies are not. And so our early predecessors were really focused on the sardine. However, why the early stage if the fishery is for adults? Well, it's because I said there's catchability, they're easy to catch. And also because most fishes in the ocean have a pelagic larval life history stage, meaning when they're babies, they're up at the surface. I'm sure we can all think of an example of a fish that lives at the bottom, maybe a halibut or a flounder, something that's flat. Or an oarfish. We don't really routinely catch those, but they do have larvae that are pelagic and aren't at the bottom. But so many species have this pelagic larval stage. And so when they're in the upper 200 meters of the water column, because their mouths are very tiny and they need high densities of food, it's up where the sun is. And so scientists could catch them. They could go out on boats and catch them and catch lots of them all the time. Salute. And so that's what was happening. That's why they said, let's go after them. But here's the thing. Inside of those samples wasn't just sardines. It was hundreds of other species of larvae. And people didn't know what, the scientists doing this work didn't yet know what those species were. And so I put up some of my favorites up here, and if there's time, maybe after we can go through them. They're so cool. They're so, so cool. Just really fast. Does my cursor work? Oh, no. All the way to the right, the second one from the right is a blob sculpin. It's a blobfish. That's a baby blobfish. So cool. Okay, anyhow. So again, all different species are being captured in these plankton jars. And so the first director of the Southwest Fisheries Science Center, Albert Ahlstrom, as you see there, he and his protege, Jeff Mosier, they together went with Ollie's vision of, yes, we are trying to answer this sardine question. We are trying to describe the early life stages of sardines. However, this jar is filled with so many other things. There's a dynamic ocean. We really should know what all the other species are, because maybe they're interacting with sardines in a way that has an impact on how they make a living. Maybe they do. And then also, Ollie said, we need to collect our samples in as quantitative a way as possible so that we can do comparison studies. And this set us up. This set us all up, Scripps, fisheries, and Cal Fish and Wildlife, to be able to do a lot of work, ask a lot of questions. And there are still more questions to be asked. And because these decisions were made early on and maintained through today, we are able to do that. Retrospective analyses are something all of us are interested in. And we'll talk a little bit more about that in just a bit. So these guys did it, and they made this decision and they stuck with it. And for you here, I have one of my favorite examples of why it takes decades to write a book like that red one that's right there, Atlas 33. I think that if you are interested in looking at some beautiful line drawings of the early life stages of the fishes that live here off our coast, you should take a look. It's publicly available on calcofi.org. And also, if you're looking for inspiration on maybe some of the crazy creatures that you read about, hear about in media, inspiration comes from these guys. Because what you see right here, this example, is an Idiocanthus. This is a Pacific black dragon. And as a larva, it looks, I would say, kind of like a slug. Its eyes are on stalks. Its body's quite long and slug-like, I don't know. And if you look in the bottom of that line drawing, farthest to the left, that's its gut. Its gut is not internal to its body. It's hanging off. And at some point throughout its ontogeny, while it's developing, it will turn into that very scary, very voracious midwater predator, which is pretty amazing. And the folks in Ollie's lab, the lab where I received my training later, they discovered that. It took 40 years to write this book, doing all of that sampling and spending countless hours at the microscope. And for that, we are indebted to these folks. They have done so much work to teach us about our fisheries and the species off our coast, because these data are utilized. Again, I said that this was a fisheries management program to begin. It has blossomed into this pretty awesome ecosystem monitoring program. But if we just look at fisheries for a moment, these data that these taxonomists resolved for us have been used in both stock assessments, like for anchovies, sardines. I'm sure you've heard of these. A couple kinds of rockfishes. Maybe some of you fish. Maybe some of you eat this type of fish, or cabezon. Maybe you know sheephead or California halibut. And then CalCOFI data have also been used in informing fishery management plans. So how these things are managed for some of these other species, like mackerel, Pacific mackerel, market squid. Who doesn't love calamari? White sea bass, spiny lobster. And then, you know, we have this wonderful example all the way to the right. We have a couple of basses. And, you know, we talk about the foundation of our program is sardines and partnership. And the partnership that we have is super special. And here's a perfect example of that. A colleague of mine was a PhD student in Bryce Simmons' lab. And Bryce Simmons is the Scripps CalCOFI director. And before Erica Mason went to Bryce's lab, she was a California Department of Fish and Wildlife scientist. And so Erica was very well versed in what CalCOFI has going on and has a very keen interest in taxonomy. And so what she did, she worked on understanding and a retrospective analysis of the samples that we painstakingly take care of so that scientists can come and ask these questions and interrogate our samples. And Erica made major breakthroughs in figuring out new ways to identify these species of larvae of these basses that are really hard to morphologically tell apart. And now those information that Erica resolved are being utilized in, these are 2 state-managed species, to help inform the recruitment questions as well as potential stock recovery, which is pretty cool. And again, this partnership, Erica started at CDFW, then went to Scripps, and now Erica's a stock assessment scientist at Fisheries, just up the hill. We love our CalCOFI family. And so again, let's zoom back out now. So we talked a little bit about ichthyoplankton ecology because it's super fun, and the babies are adorable. Now we'll go into, we'll look at our current sampling efforts again for a moment, because we want to then think about what types of data are we talking about? How are these important for us? How can we utilize them? Not just science, but all of us. What can they do for us? Because we're here to serve the people. And so these sampling efforts yield marine ecosystem data. We've talked about oceanographic data, some physics and chemistry. We are able to look at emerging climate measures. So I'm sure you've heard of ocean acidification and measurements of pH. Well, CalCOFI's been doing that and for quite a while. And we have, when you do things consistently for a while, you get a time series, and what you get is context. You get invaluable context to understand how and when things are changing. You have the opportunity to do that. We have, you know, zooplankton and invertebrate abundance, another thing. So looking at the base of the food chain for so many important predators is something that comes out of CalCOFI. And so, you know, and who doesn't love marine mammals? We get a lot of that too. And so this is just some of it. And what you'll see on this slide, and this beautiful image that Aaron Satterthwaite, the CalCOFI coordinator, put together, is that these data are publicly available. They are paid for by you, by me, by the taxpayers. They are available. And it makes it so that when things are open access, you get the best diversity of thought, and you get the best chance at really asking good questions and finding answers, because we can all come together around it. And so I talked a little bit about, you know, this retrospective analyses, interrogating samples. Well, within CalCOFI, we have 2 of the world's largest collections of ichthyoplankton and zooplankton samples. And so at the Southwest Fisheries Science Center, we retain the fishes out of those plankton samples. We sort those out and retain those. And then the plankton, the remaining invertebrates, go to the Scripps pelagic invertebrate collection. And these collections that we spend a lot of time curating and archiving and making available for anybody here and around the world. We have scientists coming from around the world, both collections, to ask their questions. And so we have things happening that we have technology for now that 30 years ago when the samples were being collected, we didn't have that technology. For instance, utilizing genetic techniques for identification. Because there are a lot of species, you've heard of cryptic species, that we can't necessarily tell apart. Well, now we have the ability to tell them apart using genetic techniques, but the folks collecting these samples 20, 30, 40, 50 years ago didn't know that was going to happen. But what they did know is that when you collect samples and you take a dip of the ocean, you better use it to the best of your ability and then shelve the rest in a way that the rest of us can use in perpetuity. And that is what happens, and we all put blood, sweat, and tears into that effort. And so I told you I would talk a little bit about environmental DNA. I'm sure some of you have heard of it before. And so if you think about it this way, when you collect a plankton sample, if you preserve it properly, if you want to use genetic techniques, the original way to do that is to take the sample and grind it up and get the genetic material out of it. Well, once you grind it up, you can't use it for anything else. Well, what was learned partly by a researcher, a PhD student working with CalCOFI, was that you can take the liquid out of the preserved sample and some of the genetics get sloughed off, just like I have skin cells that are sloughing off right now, in the liquid. And you can use metabarcoding to figure out what's in there without destroying the sample. And you can put it back on the shelf for future researchers to interrogate for additional questions. It's pretty amazing. And so our program has been collecting samples. We've been, it's another collaboration within our program, within another research program at Scripps, and it's the NOAA CalCOFI Ocean Genomics program that started in 2014. And so we have this time series of specific water samples, because water also contains sloughed off cells. And so there's lots of ways to get information out of the ocean. And we want to do that. We want to utilize it in the most sustainable way that we can. And so this is a pretty cool way to do that. And you see just here a couple of examples of how those data have been utilized to tell us what's happening in the ocean without destroying the samples. Understanding if you recall the heat wave that came in the ocean in 2014, what that looked like in fish assemblages. It was seen in fish assemblages, again, without having to destroy the fishes, which is pretty cool. Science, right? Science is pretty awesome. So now I want to get back to partnerships. I keep talking about that. And that's because it's so foundational to CalCOFI. And even though we have our founding partners, again, NOAA Fisheries, California Department of Fish and Wildlife, and Scripps, we are always collaborating. We are always working with additional partners because more together is always better. And so within NOAA, we have been, CalCOFI for a long time has been collaborating with the NOAA sanctuaries. And so this is an additional line office within NOAA. And I think I told you previously when we looked at this slide, this map, that there are regions of the National Marine Sanctuaries. And the Farallones, Monterey Bay, and what is not on this map, right around Point Conception, is now the newly designated Chumash Heritage National Marine Sanctuary, which is super exciting. This year came online, and CalCOFI has stations within that sanctuary. And we added stations, those yellow stars, if you can see them. We added additional stations just 2 years ago in 2022 because the sanctuaries has a great deal of interest in our data and have always been utilizing those data to inform their status reports and to look at around the sanctuaries. And then recently, again, a couple years ago, we worked together, CalCOFI and sanctuaries, to realize that we, CalCOFI, could provide the infrastructure and work with sanctuaries to add additional samples that are CalCOFI stations within those sanctuaries to provide information that could be utilized for their monitoring of the performance of these protected areas. And so what CalCOFI offers, even though these are new sampling stations, they are in and around the existing stations that provide this great deal of context. When you have context, you can understand and track change over time. You get to ask questions about the impacts of climate change because you can see the change over, sorry you guys, I'm not used to microphones, that scared me. Over 75 years, you can see that. It's amazing. This context offers so much. And so when we think about that and we keep moving forward, here's Chumash that you can see, we come together and can ask these questions, add more sampling, and then as we move forward together, because also within the sanctuaries are CalCOFI stations, and they're also within the wind energy development areas. Now, these areas are pretty interesting. So you see two here. One is off Morro Bay and the other one is off Humboldt. And so I definitely don't know exactly what these wind turbines are going to look like or how it's going to work specifically, but I do know that it's going to be important for us to understand the impacts of the changes that are going to happen in those areas, because it may be changing the way we utilize the ocean. It probably is. And so CalCOFI is very well poised to help answer some of those questions. Now, we already have stations in and around the Morro Bay wind energy area. Now, off of Humboldt, however, we haven't been sampling off of that area in a number of years, but we do have historical data from those areas. And so what we are working on doing is, and this is the reminder to say that we had historical stations from that area, we are now really investing, and this isn't new, because CalCOFI partners have been utilizing and working with other programs to incorporate information and data collected from uncrewed systems. But now this is different. We haven't done it off of Humboldt until last week. And so this, right off Humboldt here, that middle figure is the glider track, that little torpedo on the left there, that yellow torpedo, the glider track that was just out collecting oceanographic and imagery information. This amazing pilot survey is something that we're going to continue doing, and the glider just got picked up this weekend, and we're ready to dig into the data, because what we're doing moving forward is we're also going to expand and resume with shipboard sampling some of those original CalCOFI sites while also having the gliders out, so that we can give ourselves the chance to do some intercalibration between traditional shipboard sampling and these uncrewed systems. And so what do those data look like? Well, again, the gliders, this just got picked up on Saturday, I believe, but here's what some of the data look like. And so what you see here are some chlorophyll profiles. The green graph that's in the middle of this slide are the dives. You can see that I believe this glider dives to 1,000 meters, and you can see the chlorophyll profile that it's picking up, which gives us a lot of information about the primary productivity in the region. That's really important for understanding how everything's going to be making a living in the region. And then these images here are of invertebrates that go by the camera on the glider. And so now I don't want to paint a picture that these data are one for one can replace shipboard sampling, because they cannot. They can't do that. However, there is going to be a time when we likely can't get to all the stations that we want to be able to get to with shipboard sampling. So we are really investing in figuring out how to utilize other ways to monitor these areas so that we can be well poised to provide that context that CalCOFI always does, and help answer questions about what's happening in these offshore wind energy development areas and other aspects of change that are constantly happening. So I end on this slide because this just happened and it's pretty exciting. It's always fun to get new data, and it just hot off the presses. There's so many other things that I would love to tell you. I just tried to pick snippets that are fun. So thank you so much for this opportunity.
Thank you, Noelle. I learned a tremendous amount that I didn't know, and I've been around here for a long time. So I really appreciate that. It was a wonderful talk. We do have time for questions, so I hope some of you have put your thinking caps on. Here, I'll start right here.
Thank you so much for the wonderful talk. I'm wondering though if you can bring us up to date on the sardines themselves. Because as I gather it, every once in a while they start to grow in number, and then they relax the fisheries regulations and eat them all up again, and down they go. So how can we break this cycle?
Well, first I have to say thank you for the question, and I think you just answered it, what's happening with them. That is actually what we do observe. And so how do we break that cycle? Well, there's a couple of things. We really like to eat, people like to fish for them. They're really important, right? Well, there's another thing that's going on. There was a time in the early 2000s when our community thought, oh, we got it. We understand it now after all these years of CalCOFI and understanding the temperature fluctuations and what seemed like an opposite reaction between sardines and anchovies. We thought, oh, okay, well, when it's warm, the sardines are here. And when it's cold, the anchovies are here. Well, if you all remember in 2014, the warm blob came through and we had record landings of anchovies. I mean, they were right here off the pier in densities we hadn't previously seen. And so that's when, a lot of us got, there's this excellent paper by Francisco Chavez, and the one that said, well, sardines warm, anchovies cold. And so at a CalCOFI conference, in fact, Francisco got up on the mic and said, who was the person that ever thought that sardines liked it warm and anchovies liked it cold? Because they don't know what they're talking about. You gotta love it when somebody can poke fun at themselves. It's because more information, we realized we needed more information to see that it just wasn't that simple. And I think that's part of what the answer is. It's just not that simple. It's not a warm-cold situation. It's understanding the difference between the east and the west side of the Pacific and what we know as cold and warm anomalies rather than just a blanket statement of warm and a blanket statement of cold. It just doesn't work that way. And so that's a piece of it. And so the other piece is that we don't have a full handle on exactly how they make their living, because the question always comes back to this. It's, well, what are the exact drivers of recruitment? What makes a successful population of young ones that come back to the population, that recruit back and can reproduce to the next generation? And it's actually quite complicated, it turns out. And so I wish I had a better answer for you, but I'm gonna say the thing that a lot of us say, which is we need more data. We probably need different types of data. Maybe we don't have the right instruments yet or right technologies to measure the right things to give us the answers. And I also think it's changing. I'm sorry that was rather unsatisfying. I wish I had the answer.
Hi. Here.
Oh, hi.
I'm the chair of the Friends of the Collections at SIO.
Thank you. Thank you so much.
And I have a couple of things that tie into the CalCOFI collections. One of the things we talk about when we're trying to get people interested in supporting the collections is that when the samples are being collected, people don't necessarily know exactly how that sample will be used in the future. And there's two examples that I always tell people. One of them is, I'm sure everybody here, or most people here, are familiar with the movie The Birds, Alfred Hitchcock's movie The Birds. Well, The Birds is actually based on a true story. It Turns out that Alfred Hitchcock and one of his producers were in the Bodega Bay area, just north of San Francisco, in the 60s, and birds were flying into billboards and flying into the sides of buildings, and nobody really knew why. And then back a couple years, a decade or so later, in the 70s, the same thing happened, but there happened to be somebody doing some research off the coast at the time, and they found that there was a flagellate, a little small animal, creating a toxin, and the fish were eating the toxins, the birds were eating the fish, and the birds were going nuts. Well, some scientists here at Scripps went back into the CalCOFI collections and found samples that were taken off the Bodega Bay area when Hitchcock was there, and found that same flagellate. So there was the smoking gun as to why the birds flew into the buildings. The second one is, I'm sure most of you are familiar with the plastics in the ocean, the Pacific Gyre, where we have all these. When people think about it, they envision large pieces of plastic. They're not. They're very, very small pieces of plastic. Again, a scientist went into the CalCOFI collections and started looking at the water samples and taking plastics out of them, and was able to graph the increase in the plastics because we have something like the CalCOFI collection. And I can relate to being founded in 1949 and being 75 years old, trust me.
It's pretty awesome.
But there are more. The CalCOFI collection is an extremely important part of the entire collection at SIO. But there are other parts, and one of the things that got me involved in the collections, I've been involved since 2002.
Me too. That's when I started.
Outstanding. Right.
I mean it.
Well, one of the things that got me excited about the collections is, there are 3 biological collections and 2 geological collections. One of the geological collections is poured samples, where they take a pipe that's hollow and they drop it down into the mud and they pull it out, and they push the slug of mud out of the pipe and they lay it down and they cut it and they open it up, and you can look at the layers of sediment like the rings of a tree. And obviously the stuff on the bottom is older than the stuff on the top. And they have this one sample, and if you look at it, the layers are very, very thin. And they're gray and white and gray and white and gray and white and gray and white, and then there's this great big slug of black. And then it's gray and white and gray and white and gray and white. And if you look at the microfossils that are in these sediment layers, on the very old part, there is X number of different species. And then that number of different species gets higher and higher and higher till you get to the black, and there's nothing in the black. And then on the other side of the black, you have a few different species, not a lot, and then you get more and more and more. That black is the stuff that was thrown up when the asteroid hit the Earth 65 million years ago, and that's proof that a mass extinction happened because of that. So that kind of information is found because we have collections like that. And we need people to support the collections. We need,
Hear, hear.
We need to make people know how important these things are to science, and the bit about the DNA that she was talking about. I just recently attended another lecture about it, and they're doing it in the jungle now. And one of the primary sources for collecting eDNA is spider webs.
Ooh, cool. Thanks for being a champion of the collections.
Yeah, thank you.
Thank
very
you.
much. I have some young people over here who have some questions for you now, Noelle.
Okay, young people usually ask the toughest questions.
They do.
They do. Yes.
Unburdened by all of our
Yes.
preconceived notions.
Exactly.
My question is, what are the biggest challenges in ocean exploring, and how did you overcome them?
Oh, excellent question. For me, or in general for all humans? What? For me, the biggest challenge. That is a tough question. There's not enough time. That's the biggest challenge, is finding enough time to ask all the questions, to do all the exploring, and to also do your homework that you have to do to get to do that. It's time. It's how do you fit it all in? How do you get all the tools, all the resources? If I had all the time in the world, I could get to the right people, to get the right resources, to get the support for collections and ships and scientists and students of science. It's time. I think it's time. Time to get the resources that we need as soon as we need them.
I saw in your profile that you're a biogeochemist. What is biogeochemistry?
Oh, okay. Well, actually, I think Cheryl might want to talk about that. I am not a biogeochemist. I study larval fishes, the baby fishes. But if you want to think about the links between biology and chemistry, and then how the ocean currents move and why they do and how the earth is moving, if you link all of those things together, you start understanding how nutrients work and how they supply the base of the food web. All of these things are linked, if that makes sense to you in a most basic way.
It's a great question and a great answer. It's all one big system.
Yeah.
Okay, so what's the coolest creature that you've ever seen in the ocean?
That's a great question. The coolest creature. I don't know. There's so many. There's so many in the ocean. They're so cool. I'm a very lucky person. I've gotten to go on lots of boats and see a lot of animals. I think one of the times when we caught a pelagic octopus, and it was very alive and very happy, but we were offshore, and we needed to get back inshore to release it safely so that it had a comfortable habitat where it could do a good job making a living. Offshore, it wasn't going to. We put it in like a plastic tote, and we made a flow-through system so it could constantly have fresh water. And then we would catch plankton to feed it. It didn't like that because, well, it didn't want to eat plankton, but we couldn't very well catch crabs because they're on the bottom. But here's the coolest thing. Watching it change color and shape they're true shapeshifters. It was amazing. I've seen some other things. We've caught sharks. It's fun to catch sharks because they don't die. You gotta love that. You can catch them, you measure them, you put a towel over their eyes to calm them down, and then you throw them back. That's cool. Feeling their skin is really interesting. I've seen lots of deep sea fishes, but I don't know, seeing that octopus do its really cool puffing up and then elongating and flattening out, and just like the way that the colors were waving throughout. It's the kind of stuff that you would see in an aquarium like this one. But we were out in the middle of the ocean, and that was wild to me. It was very cool. Yeah. Okay.
So about how many of these weird creatures have you seen?
Oh, I don't know. That's a great question. I'm going to say every single research cruise, I see something pretty cool. I've been on, I don't know, 70 research cruises probably. I don't know, a lot. But every time you see something interesting, and that is something, even when the cruise is just a day, I've been on a bunch of day cruises. I've been on cruises for a little over a month. The ocean is an amazing place. When you take a net and you bring it up and you put it in a dish, rinse it down into a dish this big, and you put it under the microscope. It is a whole world that you can't see with your bare eyes, and it is fascinating. It is what gets so many of us up in the morning to go to work. It is fascinating. Every time I go to sea, I see cool things. Every time I open a sample and put it under the microscope in the lab, I see cool things. And the question I get to ask is, how do they make a living? And I get to help describe that. And then I get to use that information and help us manage our resources better. And that's pretty cool.
Well, I think that is a wonderful note to leave on. Excellent questions.
Yes, great questions.
And Noelle, thank you for your beautiful answers and for a wonderful talk.
Thanks.
So join me in thanking Noelle again. We will see
coming. Thank you. Those were great questions. I love that.
They had asked about questions.
Yeah. That was awesome.
Yeah.
That was so fun.
Excellent. That was so, I
Thank you.
learned so much. I did. That was great.
Do you know the gentleman there in the front?
I do. Well, it was the first time
He
I
was
ever
he
met
was
him
a
in person.
director.
Thank you.
He was a director when I started. I'm a member of the Directors Council.
Oh, cool.
It's a 3-year term. This is my 23rd year.
Okay. Love that.
Yeah, I do. I probably run 400 people through the collections, doing tours.
Wow, that's really good.
Yeah. Well, the first one I did. There was a gentleman in development that started the Friends of the Collection. I'm one of the founding members of the Friends of the Collections, and I'm the chair of the Friends of the Collection.
Okay.
But we decided that we needed an outreach program, and we needed to get people acquainted with the thing. And at the time, I was a member of the San Diego Council of Divers, and I was the president of the largest dive club in San Diego. So we decided if anybody ought to be interested in the collection, certainly the dive community would be. So we set up a tour of the collections for the dive